Welcome you all to this cryo-EM course, and last class I already discussed about how we can make different types of negative staining of different types of samples, and then we can image those samples. And I also discussed about the good images, bad images, and then light staining, thick staining, and the moderate staining areas. We also discuss about how those negative staining data we can use it for different purposes for our thesis and the research also. Now couple of more points we have to mention, and one of the things I didn't cover in last class, that is the DNA protein interaction and the lipid protein interaction. So today's class we are going to discuss about the DNA protein interactions and lipid protein interactions, and as well as I'll show you how to image the bacterial and mammalian cell. So here the last class I already discussed about that when we are purifying a sample that different based on the different ACC peak, we are getting different kind of oligomers. That is one of the reason in the first class also I told, first class of the purification, I also told that when you will do the ACC purification you should not mix all the fraction together, because this is the same sample which aggregates and eluted near around 8 ml in 8 ml which is basically void volume, and we are getting this kind of larger assembly where there is a 2 or 3 oligomers interacting together and forming this kind of assembly. In the other case we can see there is a peak 2 sample. In this peak 2 sample where we can see there is a nice heptamer or hexamer. This is a top view and this is the side view. Then peak 3 we are getting many monomers, which we can see there is a elongated architecture of this protein sample. Now what about if you just use that similar kind of protein in the lipid membrane. So here this is the two example where you can see this is a lipid membrane where there is some kind of bacterial proteins or the toxins they are interacting with the lipid membrane, and the same protein what I showed you in the last slide, where you can see this that particular protein, that ESPB, it is also interacting with the lipid membrane. So if you look at in this particular image, what you can see, this is a lipid environment, or those are the small lipid droplets where these toxins are interacting and forming some kind of oligomer. Here there is another lipid where several toxins are interacted there. Now what about the DNA protein interaction? I already discussed about the DNA, how to image DNA sample. Basically metal shadow we are doing and then we are imaging. But when you have a protein sample and it is interacting with the DNA sample, we are generally using that uranyl acetate or uranyl format to stain those kind of sample. Here you can see there is a very short DNA, that is 200 base pair DNA, and then we have some kind of molecules which can interact. So this is this MSDPS2 molecules. What the author did it here, that MSDPS2 molecules, it has some affinity towards DNA and it is binding with the DNA non-specific way. So when there is a 200 base pair DNA, we can see that proteins are clustering on top of the DNA. The same group, they just use that 600 base pair double stranded DNA and the same protein, where you can see this is the DNA strand, we can see it clearly. So this blue line, which one is showing, that is the DNA, and this DNA is interacting with our this particular protein, that is called that MSDPS2 protein. Here you can see there are many DNA strand where the proteins are interacting. So those kind of stain you need to we need to stain the sample very well, so DNA should be clearly visible on the background. Otherwise, if there is a thick stain or very light stain, it will be very difficult to see the DNA. You may see the protein, but you cannot see the DNA. So proper staining is very important when you want to see the DNA protein interaction. Now, in the other one, that we used a very large plasmid DNA, that is 3 kb. So now this 3 kb DNA, this 3 kb DNA is mixed with MSDPS2 protein, where you can see the DNA is completely coated with this MSDPS molecule. There we also use the another types of molecule, which is called the holiday junction DNA, where we put this MSDPS2 protein, where we can see that based on this DNA architecture, 4 MSDPS molecule interacted with that holiday junction DNA and forming this kind of complex architecture. The other one, if you just consider that there is a very large DNA, that is from double stranded DNA from 5 kb to 20 kb DNA, which is interacted with this particular protein, where you can see this is the DNA molecule, and this DNA is going through this, and then it is forming a very large fold, where most of this MSDPS protein interacted with this DNA, and then we calculated some kind of class averages from this DNA protein interaction to see what kind of interactions are there in between this protein and the DNA. Now, this is all about our lipid protein interaction and DNA protein interaction. There are many other examples are possible, but we cannot cover each and every example how that RNA protein, DNA protein and lipid protein interactions are there. So here there are some examples are there that will help you guys also to understand that how the DNA protein or RNA protein interactions are happening. Now, the next step is that how we can do the bacterial stain, if there is a bacterial sample or any other cellular sample, how we can image those sample. Most of the time we have, when you have a purified protein, it is easier to stain. Always remember that when there will be a bacterial cell or the mammalian cell, there is a high chance this bacterial cell or mammalian cell will absorb lot of staining solution, and then you will just get a very black molecules. Those molecules will be very large. Generally for the bacterial sample or cell sample, one thing we have to remember that we don't need to go very high magnification. Generally 6K to 20K magnification is good enough for bacterial cell or any kind of mammalian cell. Generally for the mammalian cell we have to go very low magnification. So when we will prepare the bacterial cell for the negative staining, how we can do that? So we should follow these basic protocols, where we will grow the bacterial cell in the media, and then a bacterial cell suspension was diluted such that the final OD become 0.2, and then cell were washed through by suspending gently with PBS buffer, and the pH will be 7.5. So we have to follow certain protocol, that pH and the pH of this PBS buffer, so it should not damage the bacterial cell. And a couple of rounds of washing is very important, because if we wash the cell properly, then there will be no other contamination will be there, because when cell is growing there will be lot of other nutrients or some cell debris and other molecules will stay. So it is better to remove those media and molecules, otherwise your overall TEM grids will be very dirty. So then cell were plated by centrifuge at a particular RPM for 15 minutes. When that process is then done, then this process you can do this process couple of round. This PBS wash also you can do it couple of round. Finally the cell pellet will be suspended with a small amount of PBS buffer, so something 200 microliter. So if you diluted with more volume, your number of bacterial cell will be less. Based on your cell pellet, you have to decide that what should be the final volume. Then you have to perform the negative staining. That is the most important part, because here I mentioned that if we just go for a normal staining procedure, what will happen, it will absorb lot of staining the bacterial cell, it has a lipid membrane, or mammalian cell, any cell it has a lipid membrane, it will absorb lot of staining solution. If it will absorb lot of staining solution, cell will appear as a very black architecture. So that is one of the reason we have to focus on this particular staining part. So as usual for like any kind of negative staining, we have to do the glow discharge of the TEM grid, and that is very important. When glow discharge is done, generally we use less than 1% uranyl acetate staining solution, and the time for staining that sample and blotting the excess buffer and staining solution, we have to do it very fast. So generally for protein sample we are using 1% to 2% uranyl acetate solution. Here generally we will use it, the solution will be less than 1%. So we have to dilute our staining solution, as well as when we will stain, we should not stain the sample more than 5 to 10 seconds, because if you do the staining for longer time, it will absorb the staining solution. But it depends on your preparation to preparation, sometime it may vary. So it is not any fixed time that only 5 seconds you have to do. Based on your concentration of the staining solution, bacteria, or that particular environment, and how you are blotting your sample, sometime you can get a very thick stain. So those parameters you may have to change it, but generally we don't use 1% or 2% staining solution when you will do a normal negative staining imaging of any bacterial cell or mammalian cell. And also this time can vary. Grid will quickly, so the most important part is the grid will quickly touch the staining solution and then blot it as quick as possible. So staining solution should not deposit. When the grid is ready, we will wait for some time, because bacterial cell most of the time it will absorb some kind of it will be hydrated for some time. So the grid should be dried completely, and then we can use that grid for TEM imaging. So let us take a look that how we can get that TEM images. So here I was showing you some bacterial cell. In this bacterial cell, this is basically E. coli cell. You can consider this is the outer membrane and this may be the nucleus, but some area it is absorbing more stain, that I exactly told you that sometime it can absorb more stain, and then it will be difficult. Sometimes some grids will be completely some cell will be completely black, that means those grids absorb lots of stain. So here there are some kind of amyloid peptides are there which can damage the bacterial membrane. So we incubated this bacterial cell with those peptides, and we are trying to monitor that is it damaging the bacterial membrane or not, and we vary this concentration of this peptide. So at lower concentration we can see those peptides can interact with the bacterial membrane here and the bacterial membrane here. It can damage the bacterial membrane, but it cannot completely rupture the cell. The similar type of image is here. Here you can see there is a two bacterial cell, they are connected, and then some kind of peptide density is appeared near the cell. So here you can see there are some kind of peptide density which is targeting the bacterial cell and damaging the bacterial cell membrane. Now if we go to the next one, here you can see there is another bacterial cell where the membrane of this bacterial cell is clearly visible at different position. So this is, I can say, it is a good stain. Also here you can see there is a cluster of multiple bacteria, and there are some peptide which is peptide or some kind of, we can consider that some inhibitory drug kind of things which is binding to the cell membrane, and there are some leakage from that cell also, and there is a morphology is changing drastically. So this kind of TEM images gives us lot of information. So this is a negative staining imaging, and it is a quick study. So at a different various concentration of the drug or various concentration of some inhibitors, if we add the bacterial cell, what is the morphology of the bacterial cell? Is this bacterial cell membrane is completely ruptured, and all the materials inside the bacteria, it is coming out. So if we want to monitor, and so starting from a very low concentration of that inhibitor to a very high concentration of the inhibitor, we can gradually increase, and each and individual sample we can image it separately, and that will give us lot of information that how this peptide is interacting with the membrane and how it is damaging the bacterial membrane. So in this particular case, we don't need cryo-electron microscopy to study this bacterial cell. We can just use normal negative staining method to visualize their morphology, and in this particular case we have a huge number of so in this particular case we can test many different concentration of those inhibitors or peptides. So in this particular case, if we try to do cryo-EM, it will be expensive as well as very time consuming. So at different time frame or so incubation of this bacteria for starting from 1 minute to 6 hours. So there will be many small sample, or starting from a very low concentration of this inhibitor to a very high concentration of the inhibitor. So at the end we may have 10 to 20 samples. So this 10 to 20 samples, if you want to do cryo-EM, it will be very difficult task. So that is the reason if we implement negative staining in this particular case, we will get lots of information, and that you can see it here, that okay, when we increase the some kind of those inhibitors, you can see in the background there is a black patches, those inhibitors are deposited here. Only we are targeting one particular cell, but in other cases you can see they are clustering because damages are more, and you can see the morphology changes in the membrane also. So if you target in this particular area of this bacterial cell, you can see more damages there. So there is a very good way and easy way to study any inhibitor concentration or different time frame, how these bacteria are behaving in presence of some kind of inhibitor. The next example is that there is another bacteria we selected. Here you can see they are forming a circular bacteria. Previous one you can see it was an elongated bacteria. Here you can see this is a circular bacteria. They are clustering because of some kind of inhibitor, and that black patches you can see, this is the inhibitor. And then if you just target one of these bacteria, what is happening here, there is a leakage from this bacteria because of some inhibitor. It is damaging the bacterial membrane, and lots of fluids and other materials are coming out from that cell, which is spreaded all over the grid. So if you image it properly, you can get lots of information here. Not only that, if you focus on this one enlarged view of one particular bacteria, you can see there is a lot of damages in different position of this bacteria, and the bacterial membrane is some areas are not very smooth. That means these are the areas where which areas are severely damaged due to those inhibitors or peptides. Here there is a one published data I just selected from the journal, that is from the JACS, where this what author did it. So they have P. aeruginosa bacteria, where this is the control cell. There is a 2 cell, and you can see the background is 10, really nice. You can see lots of details here and the morphology also. Now we took that enlarged view of that when we treated with some kind of chemical compound and want to see the morphology of the membrane, and we can see this is the morphology of the membrane. Membrane was severely damaged, and so in both the cases you can see how the bacterial shape and size is changed because of those chemicals, that is this particular chemicals. So here also that you can do a negative staining TEM images. It is a dry condition, room temperature TEM, but you can make lot of conclusion. You can get lot of useful data from this TEM images. Not only that, here I am showing you that there is alpha hemolysine is one of the bacterial toxin, and this bacterial toxin generally interacts with erythrocytes or RBC membrane. So here you can see this is a control cell, and the outer membrane of this control cell is very smooth. But if you look at this same RBC cell, when we treated with that toxin molecule, how these toxins are damaging the outer membrane. From this one you can see that there is a lot of damages in this particular area, in this particular area, and in this particular area. Not only that, smoothness of this outer membrane is completely destroyed, and there is a lot of materials is coming out from this cell membrane. Now one of the area we enlarged here to see, can we see that small bacterial toxin also here or not. Then there are many places, if we just look at those areas, you can see there is a circular toxin, and then there is a central hole which we showed here also. So from this one we can not only we can see the morphology of the cell, we can also see how it is disturbing the cell membrane, and after toxin treatment what is the actual morphology of the cell, what kind of changes is happening, and as well as we can find out some kind of protein molecules or bacterial toxins also which is interacting with those cells. Now this is the last one, last example I am showing. So far we discuss about that bacterial cell. What about the any other cell, that if you have a purified protein, and if we use that purified protein, how this purified protein is going to interact. So here you can see there are some control liposome. We imaged it at different magnification, where you can see this circular thing is very clean and very smooth boundaries also, and those are basically our liposome. And when we treated with one of our effectors protein or toxin which is binding to this liposome, and you can see here the multiple toxins are binding there. So this is the same enlarged view of this particular molecule, and this is the enlarged view where you can see individual toxin molecules. Now what we want to do, first we want to test this one, that okay, we made a liposome with a similar kind of lipid what we have in yeast, and then we tested that is it really interacting with that liposome. Now we took the yeast cell and then we incubated with the same effectors protein. So these are the control yeast and these are the experimental yeast. When we have this experimental yeast mitochondria and we incubated our toxin, so these are the liposome we made based on that mitochondrial membrane. So, this similar mitochondrial membrane we want to see it if we have a clean liposome, how it is interacting. If this is interacting properly, so we will isolate the yeast mitochondria. So, you can see this is the yeast mitochondria, and this yeast mitochondria is incubated with this liposome, and in some places you can see that circular toxin, what you can see it here on the lipid membrane. You can see similar kind of toxin is binding here. You can see two toxins are binding here. Similarly, there are several toxins are binding here, some toxins are binding here, also some toxins binding here. There are central position, you can see one toxin here. So, there are many toxins are like that. So, this is a good experiment that okay, if you look at this particular, so you can get there are some toxins are binding on the membrane. So, here you can see, here you can see there are some toxins are binding there. So, there are many this kind of experiment that you can do it using the normal temp or room temperature temp, and you can conclude this particular conclusion that okay, so that mitochondrial lipid is crucial for this toxin, where the toxins are binding on mitochondrial lipid membrane, as well as if you take the real mitochondria, toxin can bind to that real mitochondria also. So, I will finish this talk today's discussion here, and then in the next class we will discuss about the ultrasection of that cell and how do we image by the negative staining methods. Thank you for your attention.